welcome back to the deep dive. Now, if you're a medical student or maybe just a dedicated learner trying to really master the incredible engine that is the human body, well, this deep dive is definitely for you. We're taking a massive stack of sources on digestion, anatomy, and metabolism, and crafting a clear, scientifically rigorous roadmap for how we extract life from a meal.
Right.
Our mission today is to give you a deep conceptual understanding, not just the definitions, but the why behind the whole process.
And we really have to start by acknowledging the sheer scale of the operation. I mean, when you look at a sandwich or a piece of fruit, the body faces this monumental challenge.
Which is what exactly?
Reducing that physical food into microscopic usable particles, things like monosaccharides, amino acids, fatty acids. They have to be small enough to cross a single layer of cells.
And crucially, as we often remind people, the entire digestive tract is basically one long 30-foot tube that runs through you.
Exactly. Until a nutrient crosses the specialized epithelial lining of that tube, it hasn't actually entered your body.
It's still technically outside.
It is.
Okay, so let's unpack this core idea first.
Yeah.
The currency. Why do we go through all this trouble?
It's for energy.
Right, but what kind of energy?
Specifically, the energy that's required to manage the strong covalent bonds that hold all these complex biochemical compounds together.
And make and break them.
To make and break them. Life compounds, they require strong, stable structures, and that means tight electron-sharing bonds. And to power all that, cells rely on one universal, standardized currency.
Adenosine triphosphate, or ATP.
That's the one.
It's the rechargeable battery system of the cell.
It is. If you picture the APPP structure, the brilliance is really in those three phosphate groups.
Okay.
They are all negatively charged, which means they naturally repel each other.
Like trying to push the same poles of two magnets together.
Precisely. Forcing those repelling negative charges together, especially attaching that third phosphate, requires a significant energy input.
And that energy is then stored within that high energy bond.
Well, it's more that the energy is in the effort it takes to keep those repelling charges together. So when that third phosphate is cleaved off,
the stored energy is instantly released.
Instantly. And that fuels cellular work. It converts ATP to ADP, and the body just works tirelessly to recharge it. It's the engine of life. And all our food ultimately feeds this one system.
That sets the stage beautifully for the input side. So let's start with the major nutrients we feed this system, beginning with carbohydrates.
Right.
We categorize them primarily by size, correct?
That's the most practical way, yes. You have the simple monosaccharides, glucose, fructose, galactose, which are, immediately absorbable.
Okay.
Then you have the disaccharides like sucrose, which is table sugar, or lactose from milk. And finally, the really long-chain storage forms, the polysaccharides.
And that's where we get the difference between plant and animal storage. So things like amylose and amylopectin in plant
Starches, yes.
versus glycogen in animals.
And all of those are just complex chains of glucose that have to be meticulously chopped down one by one for the body to absorb.
But there's one we can't break down, and this is where it gets really interesting from a practical standpoint.
You're talking about fiber.
I am. Cellulose. What's its clinical importance if we get, zero nutritional value from it?
It serves a vital, non-nutritional role as roughage. Since it stays intact, it does two really crucial things. One, it acts as a mechanical regulator, keeping the digestive tract moving.
Right. It adds bulk.
It does. And two, and this is critical for cardiovascular health, it actively binds to cholesterol in the intestine. It actually helps eliminate it before it can be reabsorbed.
Okay, moving on to proteins. If carbohydrates are the fast fuel, then proteins are the heavy-duty structural stuff.
They're the versatile workhorses. I mean, they're essential for energy, sure, but they also build structures like collagen in your tendons or keratin in your skin.
And they function as hormones, enzymes, muscle.
Exactly. They are these long chains built from 20 different amino acids.
So how do we track if we're getting enough of these critical building blocks in a clinical setting?
We use a metric called nitrogen balance. Since nitrogen is unique to amino acids, tracking its intake and output tells us a lot.
Good.
If a person is growing or healing or repairing tissue, they are in positive nitrogen balance. Intake is higher than breakdown.
And a negative balance.
Negative nitrogen balance is a very serious sign. It means protein is breaking down faster than it's being replaced. It signals a deficiency or, major tissue loss like you'd see in burn patients.
Let's pivot to the lipids, or fats. They get a bad rap, but they are absolutely essential.
They are the long-term bank account of energy. The main storage form is the triglyceride. That's a glycerol plus 3 fatty acids.
And that gives us our insulation cushioning.
Right. The other crucial lipid is cholesterol. We make it in our liver and we eat it, and it's the structural framework for steroid hormones like testosterone and estrogen. Plus, it adds flexibility to all our cell membranes.
Now, let's get into the fat debate. Saturated versus unsaturated. What's the simple takeaway for a learner?
It really just comes down to hydrogen saturation. Saturated fats, which are mostly from animal sources, are full of hydrogen and, well, they tend to be less healthy. They're linked to plaque buildup in arteries.
And unsaturated fats.
Mostly plant-based, they're missing some hydrogens and are generally the preferred choice.
Okay, so because blood is water-based, these fats can't just float around. They need special packaging.
Yes, and this leads us right into the LDL-HDL distinction.
The good and bad cholesterol.
Exactly. Lipids are insoluble, so they have to be packaged into these lipid protein mixtures. LDL, low-density lipoprotein, is assembled in the liver to deliver fat and cholesterol out to the tissues.
And that's the bad one
Because the lipids it carries are prone to separating out and sticking to vessel walls. That's what contributes to atherosclerosis.
So what's the counterpoint? The good guy.
That's HDL, high-density lipoprotein. This particle is released empty by the liver, and its specific job is to actively scavenge excess cholesterol from tissues and, crucially, from the vessel walls themselves.
So it's like a cleanup crew.
A very effective one. It brings that cholesterol back to the liver where it's converted into bile. This scavenging action is why high HDL levels are so protective against blockages that can cause heart attacks or strokes.
Excellent. Now we've covered the majors. Let's talk about the minor but mighty nutrients, vitamins and minerals. They don't provide energy or structure, so what exactly do they do?
They are the necessary assistants. They function primarily as cofactors for enzymes. They help the cellular machinery perform all those chemical reactions we just discussed.
How do we categorize them? And what's the most critical clinical distinction between the groups?
We divide them based on solubility. You have the fat-soluble vitamins A, D, E, and K.
Okay.
And because they are absorbed with fat, they are stored in the body, and this is critical. Because they're stored, you can actually reach toxic levels if you oversupplement.
Ah, right. And the water-soluble group.
The water-soluble vitamin C and the B complex are generally just excreted in urine, so toxicity is rare. But they come with their own unique challenges.
Such as?
Well, take vitamin B12. It's water-soluble, but its absorption requires a protein called intrinsic factor, which is secreted by the stomach.
No intrinsic factor, no B12 absorption.
Exactly. And that leads to a condition called pernicious anemia.
And speaking of clinical challenges, let's mention one mineral deficiency that has really dramatic physical consequences.
That would have to be iodine deficiency. Iodine is a vital component of thyroid hormone, which regulates the body's entire metabolic rate.
So without enough iodine.
The thyroid gland swells dramatically, a condition called goiter, in a desperate attempt to capture more iodine from the blood, and that leads to a whole host of metabolic problems.
Okay, so now we have the nutrient into the cell. Let's shift our focus to what the cell does with them. Metabolism.
Metabolism is, simply put, the sum of all chemical reactions in the body. We categorize them into two buckets: anabolism, which is building things up and requires energy,
Catabolism.
and catabolism, which is breaking things down and releases energy.
And all that released energy has to be funneled into recharging our ATP batteries. We've got two main methods for that.
You do. A small amount of ATP is produced by substrate phosphorylation, which is basically scavenging leftover energy from random reactions. But the massive payoff, the vast majority of our energy, comes from oxidative phosphorylation.
And that one has a specific requirement.
It does. It requires a whole series of reactions that ultimately demand oxygen at the very end to act as the final electron acceptor.
Let's follow the primary fuel, the 6-carbon glucose molecule, through the whole glucose oxidation power chain. It starts in the cytoplasm, right?
Step 1 is glycolysis in the cytoplasm. It's a preparatory phase. Glucose is split into 2 molecules of pyruvic acid, and you get a small net gain of 2 ATP.
Then the real work starts inside the powerhouse of the cell, the mitochondria.
Correct. Step 2 is the Krebs cycle. The pyruvic acid is modified into acetyl-CoA and enters the cycle. Now, the main goal here isn't really ATP production. It only generates 1 ATP per turn.
So what's its purpose?
Its purpose is to systematically strip away high-energy electrons, producing CO2 as waste.
And those electrons go to step 3.
Exactly. Step 3 is the electron transport chain on the mitochondrial membrane. This is where the electron energy is harnessed in a series of passes to generate the massive payoff up to 34 ATP molecules.
So altogether, one glucose molecule yields a maximum of 38 ATP.
In theory, yes.
But what's fascinating here is that these aren't simple linear pipes. They all intersect.
They absolutely do. This is the body's adaptability. Amino acids and fatty acids can be diverted and fed into or removed from these cycles at various points.
Like with gluconeogenesis.
A perfect example. In low-sugar situations, the body can convert amino acids into glucose using that process. It's a crucial survival mechanism.
Now let's change gears and go back to that 30-foot tube. Let's trace the physical journey from mouth to final absorption. We'll start with the histology of the tube wall, the layers.
Okay.
But instead of just listing them, let's focus on the single purpose of the two most important layers.
That's smart. So the innermost layer is the mucosa. This layer contains the epithelium, which is that selective interface. It's responsible for protection and, of course, the actual absorption of nutrients.
And the driving force. What moves everything along?
That's the muscularis. This is a thick layer of smooth muscle with both inner circular and outer longitudinal fibers. Its sole job is to contract, generating that wave-like movement we call peristalsis.
So we enter the mouth, teeth grind the food into a bolus, and chemical digestion begins immediately.
Yes, with saliva. From the parotid, submandibular, and sublingual glands, it contains two key enzymes, amylase, which starts breaking down starches,
And lingual lipase.
which starts breaking down triglycerides.
Once swallowed, the key anatomical event is that protective reflex, stopping food from going down the wrong pipe.
That's the job of the epiglottis, which flips down to cover the trachea. When this system fails, or if the cardiac sphincter at the base of the esophagus fails, you get a very common clinical complaint.
Heartburn.
Caused by acidic stomach contents irritating that esophageal lining.
Next stop is the stomach, the specialized blender. What makes its muscular structure so unique?
It has three muscular layers, circular, longitudinal, and oblique.
Ah, the third one.
Right. That third layer allows it to constrict and churn food much more vigorously into a thick paste called chyme. Its inner lining is also folded into rugae to allow for expansion.
The secretions here are intense.
Yeah.
Down in the gastric pits, what are the three most critical components we need to track?
Okay, first, hydrochloric acid from the parietal cells. It's just indiscriminately destructive, kills most microbes.
Second.
The chief cells secrete pepsinogen. The acid activates it into pepsin, which begins protein digestion. And let's not forget, those same parietal cells also secrete intrinsic factor.
Which we already know is mandatory for B12 absorption later on. And it's fascinating that we now know the primary cause of stomach ulcers isn't just stress, but a specific organism fighting that acid.
Exactly. The main etiology is the bacterium Helicobacter pylori. It's a survival master. It burrows under the protective mucus layer and creates ammonia to neutralize the surrounding acid.
Allowing it to directly damage the mucosal cells.
Right.
From the stomach, the chyme enters the real absorption hub, the small intestine. How does this section maximize surface area?
It's an elegant hierarchy of folds. You start with the large circular folds, then projecting off those are finger-like villi,
Which contain the capillaries.
the blood and lymphatic capillaries, yes. And finally, each individual epithelial cell has microscopic projections called microvilli. It creates this massive absorptive area across the three segments, the duodenum, jejunum, and ileum.
And the duodenum receives this huge input from accessory organs. It's like a chemical factory.
It is. The pancreas delivers two vital things, massive amounts of bicarbonate to neutralize the acid, and all the key digestive enzymes, pancreatic amylase, lipase, and trypsin.
At the same time, the gallbladder is releasing bile.
Which is absolutely essential for emulsifying large fat globules.
Okay, let's separate the absorption pathways here. Where do the water-soluble nutrients go?
That's the simple path. Sugars and amino acids pass directly into the blood capillaries within the villi. From there, they drain into the hepatic portal system and go straight to the liver for immediate processing.
But the lipids are different. They avoid the liver at first. Trace that unique journey for us.
This is really the highlight of fat absorption. Bile salts coat the digested fats, cholesterol, long-chain fatty acids to form these microscopic transport spheres called micelles.
Okay.
The micelles shuttle the fats across the epithelial cell membrane. Once inside the cell, they are repackaged into much larger transport particles called chylomicrons.
And these are too big for the blood capillaries.
Far too big. So instead, they enter the lymphatic vessels, the lacteals, bypassing the liver and entering general circulation much later, up near the neck.
After all that action, we arrive at the large intestine, where the primary focus changes to, Water salvage and cleanup?
That's exactly right. The large intestine's main function is hydration regulation. We absorb nearly all the water that entered the digestive tract, leaving only a few ounces to be excreted.
But it's also a vital microbial ecosystem.
It is. The normal flora are bacterial residents. They finish digestion, fermenting unabsorbed carbohydrates, which, yes, does cause gas. But critically, they synthesize essential nutrients for us,
Like vitamin K.
especially vitamin K and some B vitamins, too.
And when this system fails, like during diarrhea, the clinical consequences go far beyond just dehydration.
Oh, absolutely. Diarrhea causes dangerous water loss, of course, but the biggest immediate threat is the loss of electrolytes, sodium, and potassium, which are vital for heart and nerve function.
It also causes a loss of bicarbonate.
Yes, which can rapidly lead to blood acidification, a very dangerous state.
Let's address some modern nutritional challenges. Our understanding of diet has evolved so much since, say, the 1990s. We've moved from that old food pyramid.
Which just focus on reducing all fat.
Right. And that shift was crucial because that old paradigm encouraged a surge in refined carbohydrates.
Which led to a major biological problem: rapid glucose spikes.
What's the specific danger of a rapid blood glucose spike?
It forces the pancreas to release high levels of insulin to clear the glucose from the blood. If these spikes are frequent and intense, a constant assault, your tissues start to lose sensitivity to insulin.
And that's a major contributor to the rise of type 2 diabetes mellitus.
It is. And, critically, it promotes increased fat storage and obesity.
So if the problem is the speed of the glucose release, the solution must involve choosing foods that release it more slowly.
And that's where the glycemic index comes in. It's an incredibly useful tool. It's a scale where pure glucose is 100, and it tells you how rapidly a food will flood your system with glucose.
So choosing low-index foods helps maintain that insulin sensitivity.
It prevents those sharp hormonal spikes that drive fat storage.
Finally, let's wrap up with two common digestive problems that really illustrate systemic failures. First, lactose intolerance.
A very common one. It's a simple deficiency in the enzyme lactase. Without it, lactose sugar remains undigested. It acts as an osmotic agent, so it draws water in, causing bloating and diarrhea.
And then the bacteria get to it.
And then it's fermented by bacteria in the large intestine, which causes gas and cramping.
And what about the broader, more serious issue of malabsorption syndromes?
These are generalized deficiencies. So if you can't absorb lipids properly, maybe due to a lack of bile or pancreatic lipase, it leads to steatorrhea, a frothy, foul-smelling stool.
And deficits in all the fat-soluble vitamins, A, D, E, K.
All of them. And if protein absorption fails, the liver can't make enough plasma proteins like albumin. Since albumin controls osmotic pressure in the blood, low levels cause water to escape the vessels and pool in tissues. You get severe edema.
So what does this all mean when we synthesize the whole journey? It's far more complex than just a simple input-output calorie calculator.
Absolutely. It means health isn't just about the quantity of calories, but the quality and how they're handled hierarchically. The difference between a refined carbohydrate and a whole grain isn't trivial. It's the difference between a controlled cellular energy release and a hormonal tsunami that threatens your insulin signaling pathways.
The entire 30-foot factory is an elegant but fragile system.
It really is.
That's a perfect synthesis. And speaking of that elegant system, let's leave you with one final thought exercise to reinforce the most complex absorption pathway we discussed today.
Okay. Think about your absorption pathways. Trace the journey of a long-chain fatty acid from the moment it interacts with bile in the intestinal lumen to the moment it leaves the epithelial cell. What unique structures, from micelles to chylomicrons, and what unique carrier system are required to facilitate its transport, and where does it first enter the general circulation, deliberately bypassing the liver?
Understanding that pathway is key to understanding the critical role the small intestine plays. We'll see you next time for another deep dive.